Before I go into the details of the vaccine, it is very important to know how does the virus enter the body and how does our immune response responds to this virus. As you all know that the virus is a has number of spike proteins, and these spike proteins, they latch upon to the ACE2 receptors, and these once the virus is inside the cell, it releases its mRNA and uses the cell material to form various copies of the virus. Once the virus is released to the milieu of the cells, and the immune response is response by recruiting the antigen presenting cells or the dendritic cells, which engulf the virus. And not only they engulf the virus, they present the peptide antigens of the virus on their dendrites. This antigen is recognized by the T helper cells, which in turn recruit the B cells to produce a lot of antibodies, which in turn neutralize the virus. Also, there is a cytotoxic T cell response, and these cells, once they are recruited, they are able to neutralize or they are able to kill the cell, which is having a number of viral copies. And there are two important things. One is the T cells over here, one is the B cells and the antibodies over here. It is not that all these T cells and the B cells are there in large number. There may be a few small number of cells, and they keep on patrolling the whole body. And whenever there is a new attack, they multiply in number and recruit larger number of cells and produce good number of antibodies. There have been different types of vaccines. Before I go into the what sort of current vaccines are there, it is important because the vaccine technology has changed since the time we read it in about 30 years back. As you all know, that the first type of vaccines are the virus vaccines or the live attenuated vaccine or the inactivated vaccines. For live attenuated vaccines are those vaccines in which the virus has been attenuated in such a way that it is able to generate an immune response, but it is not able to generate the disease. And second is the inactivated virus vaccine. Most of the virus, good number of vaccines in the current scenario belong to the inactivated virus vaccine. And the live attenuated vaccines are pretty few in number because live attenuated vaccines, they require stringent criteria because in case the virus goes in such a manner that it is able to produce a disease, it may create havoc. But these vaccines, they have a limitation because the large number of viruses are required to be cultured and produced. So our Serum Institute of India is collaborating with US-based companies called Coda Genix to produce a live attenuated vaccine in which the code of the genetic code of the mRNA has genetic code of the virus has been changed and it has been attenuated in such a way. The virus in such a way that it is not able to generate the disease. The virus is still in the phase 1 or the preclinical phase rather. Inactivated virus vaccine, India is already in the upfront by the way of Bharataya vaccine. Now we have the protein-based vaccines. As I said earlier, that the virus vaccines are slightly difficult because it requires good number of viruses to be cultured to produce these vaccines. So we have come to the component-based therapy, and we are talking about the spike protein-based vaccines. Why spike protein? Because you all know that the virus surface has number of spike proteins. These are spike proteins which latch upon the ACE2 receptors, and if we form the antibodies against these spike proteins, we might be able to prevent the attachment of the virus to the receptors, and then thereby we can prevent the disease. The protein subunit vaccines are the largest number of vaccines being produced in the world all over, and a lot of work has been going on in various countries about these vaccines. Not only the protein subunit vaccines, rather the part of the protein, which is also called the virus-like particle vaccines, are being worked upon. It is quite possible that this part of the vaccine, this part or the fusion part of the vaccine, fusion molecule or the spike protein is used. And this protein might be produced outside the body in the laboratory, or this may be produced inside the body by various mechanisms. And this can be used to be injected in the human body to produce the antibody response. And these proteins-based vaccines are pretty difficult to manage because to produce the subunit, the spike protein, and to purify it is a slightly difficult task. And that is the reason that we have not been able to have a vaccine of protein-based spike protein-based vaccine on board till now, despite other vaccines being able to come to the surface. Now we have the viral vector vaccines. It is very important because there are quite a few vaccines which are there in the market. And what is viral vector vaccines? A good number of teams have been working in this front, and in which the viral mRNA, which codes for the spike protein, is integrated into the DNA of a virus, which acts as a vector to take this mRNA into the human body. And this virus is generally an adenovirus, which can be a chimpanzee adenovirus or this is a human adenovirus. Why adenovirus? Because adenovirus is potentially harmless viruses. And this virus can be a non-replicating virus. Most of the vaccines on board are non-replicating viral vaccines because the code DNA of the virus is changed in such a way that it is not able to replicate itself. Once the virus is not able to replicate itself, whatever the viral particles we inject into the human body will produce the spike-based proteins, but this virus will ultimately die. And we'll have to inject another second dose of the vaccine as a booster dose. And the replicating viral vector vaccines, in which the virus which is injected inside the body is able to replicate, and the second dose of the vaccine is not required. The J&J vaccine was a vaccine similar to this, but this too has gone into trouble. The problem with these vector-based vaccine is that in case the human body already has the antibodies against the vector, that is the adenovirus, then it can cause issues with these vaccines, and these vaccines might turn to be ineffective. And that has been the reason that the Oxford-based vaccine or the China-based vaccine, which I'm going to talk recently, have become suddenly non-popular, and they have not been found to be very effective. And third, the most popular one is the nucleic acid vaccine, the DNA vaccine and the mRNA vaccine. mRNA vaccine is one vaccine which is pretty fast to develop. And you might wonder, why am I saying that the mRNA vaccines are pretty fast to develop? Because you might have heard that the RT-PCR based testing, the mRNA is separated. It is pretty easy to separate the viral mRNA. And mRNA can be encased into the lipid nanoparticles. And these lipid nanoparticles, they can be injected into the human body. And they once fuse with the cells, they start producing the spike proteins, and the body develops an immune response against them, and we are able to have a good immunity, neutralizing antibody and the other binding antibodies. DNA vaccine is another similar kind of vaccine in which the mRNA is integrated into the DNA of a virus, and this whole DNA is introduced into the human body by some handheld device which is called as percolator, and this DNA starts manufacturing the spike proteins. And you might be wondering that how India is doing all this. India is working upon all types of vaccines: live attenuated vaccine, inactivated virus vaccines, mRNA vaccines, vector-based vaccines, the protein subunit vaccines, all types of vaccines India is working. And why does a country have to work upon all the types of vaccine? Because vaccines are known to fail. Vaccine development has been a pretty long process, and it has been taking nearly 20 years. And this 20 years to 10 years of time has been reduced to 10 months. And for the first time in the history of the mankind, we have been able to have a vaccine on board within a 10-month period. This has been able to, we have been able to achieve because of various reasons. The whole world has got united. A lot of huge investments have been done, and the approval process has been made pretty fast. Let's go into the details how various vaccines on board are using which sort of technology. This is the data which was downloaded yesterday, and we have nearly 233 vaccines on board, out of which about 61 vaccines are into the clinical phases, and about 172 vaccines are into the preclinical development. Most of the vaccines, as I said, they are trying to use the protein subunit vaccines, and next popular are the viral vector vaccine, then is the DNA vaccine, and then is the mRNA vaccine, nearly head to head. This is about the statistics, and this is the statistics about the clinical vaccines, the phase 1, phase 2. One vaccine has been abandoned. This was one in the Australia. And these are the various vaccines in the various phases. You will see that the Pfizer vaccine, the BioNTech vaccine, which is mRNA vaccine. I said the mRNA vaccines are pretty fast to develop. And this is the proof over here. We have the mRNA vaccine on board within 10 months because we are able to separate the mRNA and inject it into the human body. And the adenovirus vaccines, again, the vector-based vaccines are again very fast to develop, but they develop a lot of issues because of the existing antibodies or the development of the antibodies by the human bodies against the vector. We'll be talking about them later. And third is the protein-based vaccines. Novavax, again, a very good vaccine, but we have not been able to bring it on board because the outside the human body production of spike protein in itself is a very difficult process because purification of the protein and then to add the adjuvant into it so as to enhance its immunogenicity is a pretty difficult task. Now, what are the vaccines in India? As I said, that all types of vaccines in India are being developed. As I said, it is the first is the AstraZeneca vaccine or the Oxford vaccine, which is also called as the Covishield. It is being used in 2 doses and it is currently in phase 3 trials. And second, we have the Gamaleya vaccine or the Russian vaccine is called as Sputnik vaccine. There are 2 doses being used on day 0 and day 21. And it is also again in phase 3. Both these vaccines are in phase 3. And then we have the Novomax Vaccine, which is a wonderful vaccine. And this is also in phase 3. Then we have the Inactivated Vaccine or the Bharataya vaccine. It is also in phase 3. Phase 2 trial data has been published. Then we have the viral particle-like vaccine, in which I said that it is being developed by the Serum Institute of India with another company. And then we have the live attenuated vaccine, the Codagenix and the Serum Institute of India. And you might be wondering to know that by knowing that even the mRNA vaccine is being developed by an institute based in Mumbai, it is the Genova Institute, and it is in the preclinical phase. So we have all sorts of vaccines on board. Which vaccine is likely to get a successful result, produce good amount of antibodies, it is a time to, only the time will tell. Now, first I'm going to talk about the Bharataya vaccine. It is an indigenous inactivated virus vaccine. Bharat Biotech and Serum Institute of India are two important bodies in the world, and about nearly 2/3 of the entire world children are vaccinated by the vaccine produced in India. They have the large vaccine manufacturing facility. They not only have the large manufacturing facility, they have one of the biggest manufacturing facility in the world. And whatever vaccine in the world cannot succeed without the contribution of the India because of its mammoth manufacturing facility by Bharat Biotech and the Serum Institute of India. This is the result of the phase 1 and phase 2. For the phase 2, nearly 897 people were recruited. These are the recruitment criteria. And after the exclusion, nearly they were randomized to 375 people. And this is the data of the side effects. And initially, there were some side effects, the pains, and local side effects, and there's some systemic effects or side effects also, fever, body ache, fatigue, headache. And you will see that they were mainly common after the first dose. They were hardly seen after the second dose. And the vaccine has been used in 3 microgram dose and the 6 microgram dose with the L-gel, that is the LM, which is used as an adjuvant, and IMDG. IMDG is again a adjuvant, and adjuvants are the substances which are added to the vaccine to enhance its immunogenicity. And this is the data which shows that the vaccine has been able to produce good antibody response. And this is the neutralizing antibody response to the vaccine, and good amount of antibodies, even the neutralizing antibodies have been produced by the small dose as well as the large dose when the adjuvant L-gel IMDG is given along with the inactivated virus. And this is the data which is regarding the CD4 cells and the CD8 cells. That is, we have been able to demonstrate a long-term memory in the form of T cell response, and there have been a good T cell response. And the vaccine, the Bharat Biotech vaccine, has been compared with the convalescent serum, which is a gold standard in any vaccine development, because the convalescent serum development of the antibodies and the helper T cells is one thing which we want actually to happen. Vaccine is one thing which is able to produce the antibody response like that of disease without causing the disease. So this vaccine is one of the promising vaccines, but it is likely to take another few months because the phase 3 trials are still underway. The conclusion of the paper, research paper submitted, says that the Bharat Biotech vaccine or the Covaxin BBV152 induced binding and neutralizing antibody responses with the inclusion of L-gel IMDG adjuvant. They have also been able to introduce a Th1 based response. This vaccine is likely to be available to the masses somewhere between March and June 2021. And because this vaccine has a cold chain requirements which are compatible to the other immunization programs already being held in India, it can be stored between 2 to 8 degrees centigrade. This vaccine is likely to be available to the masses because this is an indigenous vaccine, we have the large production facilities, we have a good antibody response, and it can be stored at the available cold storage channels. So this is the likely vaccine for the mass application in India in case the phase 3 trials succeed. Now we'll be talking about another vaccine, or the AstraZeneca vaccine, or the Shadox-1 vaccine. This is the one vaccine which was named as Shadox-1 because it uses a chimpanzee adenovirus, and it is being produced by the Oxford University. And this vaccine was, this is the trial, this is the results of the phase 1 and phase 2 trials. This vaccine, the important thing and the unique thing about this vaccine was that, and as the placebo, we use the meningococcal vaccine. Why a meningococcal vaccine was used as a placebo? Because the investigators did not want to bias the results, because if only the saline was used as the placebo, there will not be any reaction in the body in the form of fever, local heat, or local symptoms. And the person receiving might be very categorically say that he has not received the vaccine, and his behavior or the observer's bias might creep in. So this vaccine, the placebo, was given as the meningococcal vaccine. And you can see the local reaction. The important thing is the lesion, because this color, the yellow color, shows the mild symptoms, and the dark color shows the severe symptoms. You can see hardly any red colors. They have been mild in duration, age, pain, redness, swelling, tenderness, warmth. And in the patients who have been receiving, and they all responded to the paracetamol. Systemic reaction in the form of chills, fatigue, fever, feverish, headache, joint pains, malaise, etc. were also pretty mild, and they occurred in the initial 1 or 2 days. And there were hardly any severe reactions or hospitalization. So what does this data indicate? This data indicate that the Shadox vaccine or the AstraZeneca vaccine is a fairly safe vaccine. There is no issue about the safety of this vaccine. And there have been some local reactions. There have been no serious adverse events related to the vaccine. Now let us come to the the most important, that is the efficacy of the vaccine. The antibody response to this vaccine. The vaccine showed an efficacy of 62% in the patients in the form of neutralizing antibodies, as well as in the form of binding antibodies. 62% in the current scenario is a pretty low antibody response. And the WHO describes a vaccine to be called as a vaccine only when the antibody response, the efficacy of vaccine is more than 50%. And a vaccine is to be called as a good vaccine only once the vaccine is effective more than 70%. So as per the WHO criteria, the vaccine is not a good vaccine. How all the goof up with the phase 3 trials of the AstraZeneca vaccine came into the picture. The phase 3 trials were going on in the Brazil, where about 3,000 people were vaccinated, and the usual first and the second dose were given, and was found to be nearly 62% effective. In UK, 12,000 participants were given the vaccine. When the vaccine data came to the surface, the company said that by mistake, their contractor has decreased the dose of the first dose of the vaccine, and by giving half the first dose of the vaccine was able to produce about 90% response. The world has not been able to swallow this theory of the company, and there have been a lot of criticism that how could the contractor reduce the dose. But not going into all that, how do we scientifically substantiate the fact that decreasing the first dose can enhance the antibody response? As I said earlier, that the vaccines, they can produce antibodies against the vector. And this is a chimpanzee adenovirus vector vaccine. It is possible then that when we gave first dose as the half dose, the antibody protection against the viral vector was reduced and vaccine was able to mount an antibody response. But what is the truth? We do not know. As of now, the vaccine has not been granted any approval anywhere in the world. But India has lot of things on the stake about this vaccine, because Serum Institute of India has been in collaboration with the Oxford AstraZeneca for the large scale production of this vaccine, and the vaccine is available in large quantity, though it does not be released for the public consumption and will not be released till the time we have the good efficacy data. But how to go about it? It was decided, it was rather offered by the Gamaleya Institute that both the vaccines, if they're given combined, are they able to mount a better antibody response? And this is likely to be the trial in times to come, because Gamaleya Institute, as I will say, the Gamaleya vaccine, the Sputnik 5 vaccine, is also a combination of two vaccines. It is possible then that when we combine two vaccines, which are using different vectors, we are able to mount a better immune response as against when we use only one vector. What happens in the future, only the time is going to tell. Now we come to the Ad5 vector vaccine, that is the Chinese vaccine. This vaccine, when I presented my data way back in April or the May, was the front runner vaccine. This vaccine uses adenovirus 5 as a vector. And this vaccine, the China was feeling very proud of this vaccine. But again, this vaccine has fizzled out because it could not produce a sustained immune response, and the phase 3 trials of this vaccine could not kickstart in Canada because it had a variable response, particularly in the older age people and the people more than 55 years. Antibody titers and seroconversions were pretty low. Why in the old age the vaccine showed a low antibody response? Because probably these people already had antibodies against the vector adenovirus 5. As of now, the China is using this vaccine on its military personnel, but this vaccine seems to have gone out of the board, and not much is being talked about this vaccine. Now we have another vaccine in India, that is the Sputnik vaccine. The Sputnik 5 is a Russian vaccine which has been developed by the Gamaleya Institute, as you all know. This vaccine is a combination of two vaccines. Why two vaccines? Because first dose adenovirus vector 6. As I said, the Oxford vaccine was a chimpanzee adenovirus. This is a human adenovirus, and rather human adenovirus, there are two viruses are being used, adenovirus 26 and adenovirus 5. And different doses of vaccines are being tried, and 2 doses are being given on first dose on 0 day and second dose on 21 days. Why two different factors? Because in case as the first factor is able to mount an antibody response, and even if the body develops antibodies against the vector, the antibody response is already there, which is boosted by the second dose of the vector vaccine. And this vaccine has been approved by the Russia for its limited use, and you can see that there was some systemic and local reaction across the studies, pain in injection site, hyperthermia, headache, asthenia, muscle joint pains. Majority of the people had mild symptoms, and there were no major adverse events. And this is the binding antibodies, and this is the neutralizing antibodies. And this vaccine also has been compared against the convalescent serum, again a gold standard. And we see that there's a good number of antibody titers for the binding antibodies as well as for the neutralizing antibody. This too is a good vaccine on board, and India has already placed about 200 million doses of this vaccine, by which we might be able to vaccinate about 10 crore people. And you all might be aware that the Reddy's laboratories, they have been conducting phase 3 trials in India, and the Hetero Pharmaceuticals will be having the mass manufacturing facility of this vaccine, and the Mankind Pharmaceutical in India is going to have the distribution rights of this vaccine. Now we come to the Novomax vaccine. It is a nanoparticle vaccine, as I said, that the spike protein, the part of the spike protein, they can be used to mount the antibody response. And this is Novavax is basically an American company, which again has collaborated with the Serum Institute of India for the mass production of its vaccine. And we are likely to produce around 500 million doses of the vaccine by mid 2021. This vaccine, again, a number of doses have been tried. The special thing about this vaccine is that the company has been able to add a matrix 1 adjuvant. These vaccines, as I said earlier, that all the protein-based vaccines, they need an adjuvant to enhance the the antibody response. And this matrix M1 response is their own patent adjuvant. I told you earlier that in the Covaxin or the Bharataya vaccine, they have used alum or AMDG as the adjuvant, but here it is the matrix M1 adjuvant. And the vaccine has been tried in the people between 18 to 59 years of age. Different doses of the vaccines have been tried. And the vaccine has been found to have very little side effects. And it's a wonderful vaccine. If you go to the IgG response, we find that there's a huge IgG response. This is the maximum IgG response seen with any vaccine on board. And their IgG response has been nearly double that of Pfizer vaccine and about 6 times that of Mrna Vaccine. But the phase 3 trials of the vaccine are going on way. And how does the vaccine behave? We have lots of hope with this vaccine, but only time will tell how does this vaccine behave in time. We are likely to have about 500 million doses of the vaccine by mid 2021 and about 1 billion doses, more than 1 billion doses of the vaccine by the year end. And as I said earlier, this is the vaccine of the future for most of the Indians. Why different doses of the vaccines are important? Because old age people, we might have to give higher dose, because the older age people, they have decreased immune response, and unless proper dosing and different doses are tried, some vaccine studies might fail. Now we come to the talk of the town, that is the mRNA vaccine. First, the mRNA vaccine, I'm going to talk about the Mrna Vaccine. Mrna Vaccine trials have been done in USA and other countries, and the Mrna Vaccine has been approved by the USA, and I've been told that it has been approved by the UK also. And Mrna Vaccine, again, is an mRNA vaccine, and mRNA of the virus is encased in the lipid nanoparticles, and these lipid nanoparticles, they are injected into the human body, and the human body starts forming these spike proteins and starts mounting the antibody response. And once this vaccine was injected, you see that the reactogenicity of this vaccine was mild. There were hardly any severe symptoms in most of the participants. And so the safety of the vaccine is pretty safe vaccine. And these are the antibody responses. And in the binding antibody responses were excellent. And there was again compared to the convalescent serum, and is found to be a wonderful vaccine. And same the neutralization titers. And it was comparable to the convalescent serum. So fairly good vaccine on board. Now we come to the Pfizer vaccine, which is the talk of the day, and everybody in the world wants to have a Pfizer vaccine. But are we in India likely to have a Pfizer vaccine? And it has been given an approval all across the world. The UK, USA, Bahrain, UAE, and many other countries have given the approval to this vaccine. Let us see what is this vaccine. The Pfizer BioNTech vaccine. BioNTech is basically a biotechnology company which had developed this vaccine, and they have collaborated with the Pfizer for the world distribution. So mainly a BioNTech vaccine. The BioNTech has a specialty in managing the mRNA, and they have been doing it for the various cancer patients. They have been able to mount the gene therapy for the various cancer patients like melanoma, multiple myeloma, acute myeloid leukemia, where they have been able to produce antibody responses against the myeloma proteins and other cancer antigens. And they had the experience, and they utilized their experience in producing the vaccine for the coronavirus. And they 4 vaccine candidates they had, and they had modified the mRNA into 4 way 162A1, 162B1, and B2, and they used these two 162B1 and B2 mRNA, and they mounted a vaccine against. And you find that they use basically the 10 microgram, 20 microgram, 30 microgram, and this was the only trial in the world which was done in people more than 65 years of age. Most of the vaccine trials have been done in people up to 55 years of age, and this was wonderful trial. The data is robust. They had a wonderful antibody response, and these are the serum neutralizing titers, and they have been compared against the human convalescent serum, and you find that the neutralizing antibodies have been really wonderful. And not only the neutralizing antibody, they have a strong T cell responses also, and you can see that these interferon-based CD4 cells and CD8 cells, and they have been really encouraging. And this is the phase 3 trial data. And now I'm going to give you, this is the active ingredient. They have the modified mRNA, they have the cholesterol, they have the potassium chloride, potassium biphosphate, sodium chloride, sucrose, and other things. These are the components of the vaccine. And if we compare this vaccine head to head, the 2 mRNA vaccines which have been approved, the Pfizer BioNTech vaccine has been used in more than 43,000 people in phase 2, 3, and Mrna Vaccine has been used in more than 30,000 people, and both have shown an efficacy of 95%, which is a fairly good and a robust trial. Thank you very much, and for your patient hearing. I hand over the mic to Dr. Pankaj Chaudhary.